believe the Metab trainees have just a little announcement, right?
Yep, thank you. So just a reminder to the trainees that after Mirei's talk, we'll be having a short coffee hour / happy hour, whatever you want it to be. Mirei, I can personally attest, Mirei is an amazing mentor. She's also a member of the ATVB Early Career Committee. So I'm sure she'll have some interesting perspectives and great advice to give all the trainees. So again, that's happening after her talk, and I'm looking forward to it. Thank you.
Awesome. Okay, so I'm very excited to present Mirei, who I've known for a long time. We're both at the University of Ottawa Heart Institute, so we've been colleagues now for quite a few years. Mirei did her PhD at the University of Ottawa. She then did a postdoc at NYU with Catherine Moore. And Mirei has been obsessed with all things cholesterol since maybe infancy. It's hard to know. She really is, a fascinated scientist with all things cholesterol. So her lab has been working on some really, really cool stuff about lipid droplets, which are not just these, organelles sitting around the cell. They're actually very cool and very, very dynamic. And she's done some really pioneering work in this area. So Mirei, take it away. Thanks again for joining us.
Okay, well, thank you so much for having me, and thanks for organizing, Ibru, Katie, and Lali. This has been really a great seminar series, and I'm really glad to be part of this community. So as Katie said, I'm a little bit obsessed with lipids, particularly lipid droplets, and today I'll talk to you about lipid droplet metabolism in macrophage foam cells. So first, I'd like to give credit to two grad students in my lab that did most of the work that I'll be presenting today, Sabrina Robichaud and Garrett Fairman. So this is a bit of a vintage picture. It was taken a long time ago, back in December 2019, when we were able to get together in person and drink beer. So it's possible I may have to frame this picture because it'll likely be the last maskless picture of my two first graduate students together. So yeah, I just want to give credit to these two for leading the work that I'm presenting today. So what are lipid droplets? Well, lipid droplets are ubiquitous cell organelles, and their primary function is to store lipid. This is the traditional view, but I'll be talking about more functions that lipid droplets have because they are highly dynamic and involved in lipid metabolism. But this is the structure of the lipid droplets. They're comprised of a neutral core containing triglycerides and sterile esters, and they are surrounded by a phospholipid monolayer. So this is in contrast to other organelles in the cells that are surrounded by lipid bilayers like lysosomes, endosomes, etc. And in fact, lipid droplets are kind of like the cell's lipoproteins. So lipoproteins are also comprised of a neutral lipid core surrounded by a phospholipid monolayer and have the presence of these structural proteins. So in the case of lipid droplets, the most abundant structural proteins are of the perilipin protein family. There are 5 members: perilipin 1, 2, 3, and 4. Most people know perilipin A or perilipin 1 because that is the major lipid droplet coprotein in adipocytes. But in macrophages, you'll hear me talk more about perilipin 2 because that is the most abundant lipid droplet coprotein in macrophages. Also, there are several enzymes that associate transiently with lipid droplets. These could be lipases. So there are several neutral lipases that are responsible for neutral lipolysis. So they access the lipid droplet lipid core and hydrolyze away the triglycerides to generate free fatty acids, or they hydrolyze the cholesterol esters to generate free cholesterol for efflux. So there are a lot of known roles for lipid droplets in activated immune cells. So here are some of them. This is a figure made by my grad student, Dominique, summarizing the roles of lipid droplets in activated immune cells. So in polarized macrophages that are pro-inflammatory. Typically, we observe an accumulation of cytoplasmic lipid droplets as these cells preferentially utilize glycolysis to fuel their effector functions. In contrast, in M2 or anti-inflammatory macrophages, there are few lipid droplets as these cells preferentially utilize the lipids, the free fatty acids, to fuel fatty acid oxidation and serve their anti-inflammatory functions. So we know that some of the fatty acids come from LDL and that CD36 is important to internalize free fatty acids for beta oxidation in M2 macrophages. And also lipophagy can promote the breakdown of lipid droplets as well as neutral lipolysis to provide more free fatty acids that could sustain mitochondrial free fatty acid oxidation. There are known pro and anti-inflammatory roles for lipid droplets, as these are the source of several lipid mediators or lipid signaling molecules, for example, eicosanoids. And lipid droplets have been proposed as cell signaling hubs to promote cell proliferation. Also, lipid droplets are really important in the response to pathogens. And so typically upon pathogen infection, there's a surge in lipid droplet biogenesis. And in some cases, this lipid droplet biogenesis is good, and in some cases it's bad. So there's a lot of active research in this field. Also, lipid droplets have known roles in antigen cross-presentation and immune signaling, particularly through the toll-like receptors. So far from being inert, simple storage organelles as we classically view them, because they're quite abundant in white adipose tissue, as we know that is the storage site of lipids in the body. But now it's increasingly recognized that lipid droplets can actually, in fact, accumulate in several other tissues in response to pathogenic states, for example, in tuberculosis infection, where we see a lot of foamy alveolar macrophages. So these macrophages that are infected with the mycobacterium accumulate a large number of cytosolic lipid droplets. And this is one of the things that I was working on when I was a postdoc in Catherine Moore's lab. And we found one of the mechanisms by which MTB increases cytosolic lipid droplet accumulation in foamy macrophages of the lungs was through upregulation of this microRNA, miR33, and its sister strand, miR33 star, that repress genes involved in autophagy. And that in that sense, the bacteria reduced autophagy of bacteria and of lipid droplets, and this promoted the accumulation of lipid droplets in these cells. Also in aging, in the central nervous system, it's now recognized that there is a progressive increase in lipid droplets, and these accumulate in microglia in aged individuals, and to some extent in astrocytes as well. So following injury, such as ischemic injury that occurs during stroke, there is a release of toxic fatty acids by hyperactive neurons, and these can be taken up by the astrocytes and stored or neutralized in astrocyte cytosolic lipid droplets. So this is really also an exciting field of research in lipid droplet biology. So we still have a lot to understand about lipid droplet biology. So we're interested also in the foam cells that accumulate in atherosclerotic plaques. So we know, I mean, this audience well knows that plaque macrophages are really lipid-laden cells that take on a pro-inflammatory phenotype that drives the progression of atherosclerotic plaques. And now we also recognize that smooth muscle cells can become lipid-laden, and not much is known at all about the lipid droplets in these smooth muscle cells. So that's another interesting topic as well that our lab is interested in. But we know that the pro-inflammatory foam cells in the plaques express less ABCA1, and they're not good effluxers, and also that autophagy becomes progressively defective in atherosclerosis. On the other hand, the work of Ed Fisher's group and others has shown that there is possibility to shrink these plaques, known as atherosclerosis regression, so going backwards, and this involves an important role for anti-inflammatory macrophages, and these anti-inflammatory macrophages express higher levels of ABCA1, and so the two key contributing factors to really reduce atherosclerosis and also potentially promote atherosclerosis regression are the resolution of inflammation and the removal of cholesterol from the plaques. And so those are two key processes that are anti-atherogenic. And we're interested in the process of cholesterol removal from foam cells through efflux in a process called reverse cholesterol transport. So this begins with cholesterol export from foam cells in the peripheral tissues. So first, actually, the cholesterol must be mobilized from cytosolic lipid droplets as only free cholesterol and not cholesterol esters or not lipid droplets can efflux out. So first, the cholesterol from the lipid droplets needs to be liberated through hydrolysis of the lipid droplets. And then this free cholesterol can move out to cholesterol acceptors in the bloodstream, such as nascent HDL, mature HDL, and return to the liver for excretion into the bile, or also join the feces through transintestinal cholesterol efflux. And so this process has been known for a long time to be highly anti-atherogenic. And this is, when we talk about HDL, it's not simply a matter of raising HDL, but we need the HDL function through this process of reverse cholesterol transport. And so here, so I want to highlight the take-home message of this slide is that the first step of reverse cholesterol transport is mobilization of this free cholesterol stored in lipid droplets and foam cells. And this can be mediated by two processes. So first are neutral lipases. These are cytosolic lipases that hydrolyze the cholesterol esters in the lipid droplets to generate the free cholesterol. Or the second process is autophagy that can sequester lipid droplets and deliver these to lysosomes for lipolysis. And in macrophage foam cells, my doctoral work with Yves Marcel demonstrated that, in fact, these two processes equally contribute to lipolysis in macrophage foam cells. So 50% of the lipid droplet cholesterol for efflux is mobilized through the action of neutral lipases, and the other remaining 50% is mediated through autophagy. And this is the process that I'll be talking more about today, because not much is understood about the catabolism of lipid droplets through lipophagy and the mechanisms of this. So the hydrolysis of lipid droplets through lipophagy involves the formation of an autophagosome that sequesters the lipid droplets inside, and then these autophagosomes can go on to fuse with lysosomes and therefore deliver the components within the autophagosomes, in this instance, a lipid droplet, to the lysosome lumen, where we showed that lysosomal acid lipase is really important to hydrolyze the associated cholesterol esters. We showed that modified LDL, all forms of modified LDL, such as acetylated LDL, oxidized LDL, or aggregated LDL, can induce autophagy in the foam cells. So upon treatment of cells with these modified LDL, but not LDL, autophagy is activated and lipophagy ensues to promote the efflux of cholesterol. And we showed that the process is primarily dependent on ABCA1 ABCA1 transporter. So this transporter is really, it cycles, it shuttles through inside the cell in the endosomal compartments. And so that is not a surprise then that autophagy, okay, sorry, I'm like having issues with the flicking slides. So yes, so take home message right here is that ABCA1 is the preferred cholesterol transporter to efflux cholesterol that's liberated through lipophagy or autophagy. Now, today I'll be focused on this part of the pathway, which is I'll tell you a story about how we determined that lipid droplets are ubiquitinated for autophagic degradation involving these two proteins, AUP1 and UBE2G2, and that this ubiquitination of lipid droplets allows for the recruitment of autophagy receptors, OPTN, P62, and BR1. And this, along with other components of the autophagy pathways, such as HMGB1 and RAB protein 2, promotes the autophagic degradation of these lipid droplets. So when I started my lab at the University of Ottawa, really the big overarching question was, well, how is lipophagy? What are the mechanisms of lipophagy? And first, you should know that there are three autophagic pathways, three main pathways. So the first one being macroautophagy, which, as I touched upon, involves the formation of autophagosomes that sequesters cargo, and that this cargo is then delivered for lysosome degradation by the fusion of autophagosomes with lysosomes. There's also a second process called microautophagy, and this involves the direct internalization of either proteins, substrates, or organelles directly in the lysosome through invagination of the lysosome membrane. And then the third type of autophagy, which particularly pertains to the degradation of protein substrate, like single proteins that are recognized by chaperones and a chaperone complex, and then directly go through this transporter into the lysosome. So that's called chaperone-mediated autophagy. So in the case of macroautophagy and microautophagy, this can either occur in bulk or selectively. So in bulk, meaning if you were to take a snapshot of an autophagosome at a given moment, it would be perfectly reflective of what is inside the cytoplasm at that given time. So meaning there's no selective enrichment of a specific organelle or cargo, it's just occurring in the cytoplasm in bulk. So that would be, probably occurring to like heavy stress where we just need autophagy to degrade and recycle components to provide energy for the cell in times of need, such as starvation. In the case of selective autophagy, this involves specific enrichment of the cargo to be degraded. So in the case of mitophagy, this cargo is preferentially mitochondria. In the case of ribophagy, the cargo is preferentially ribosomes. And in the case of agriphagy, this is preferentially aggregates. And then, of course, we are interested in lipophagy, which would be the selective degradation of lipid droplets, although prior to our work, this had not been shown to be a selective process. So the selective autophagy pathways described to date involve selective autophagy receptors, such as P62, which is actually commonly found in all types of selective autophagy described to date. Also, a common one is NBR1. And then there's a host of selective autophagy receptors combinations and factors that will be specific to each type of selective autophagy. In the case of mitophagy, you see that ATG32 is important for mitophagy, but not for the other types of autophagy like agriphagy or pixophagy. And In the case of Pixophagy, ATG36 is specific to this type of autophagy. But in the case of lipophagy, the selective autophagy receptors and factors that mediate this process are not well understood and unknown. So that's what we set out to find. So we first began by, another last thing I should mention is that commonly, so the cell requires marks on the organelles to be degraded to know which organelle to be degraded. And typically the most common tag is ubiquitin. So we first asked whether autophagy in macrophages was selective. And so we looked for ubiquitination. And what you can see here is, so first, these are aggregated LDL loaded bone marrow derived macrophages. And these accumulate a large number of cytosolic lipid droplets shown here by Bodepi staining in blue. And you can see the association of LC3 at the surface of these lipid droplets, indicating lipophagy is active in these cells. Also, we observed localization of P62 at the surface of the lipid droplets. And so then we found that indeed there was ubiquitination of the lipid droplets. You can see several of the lipid droplets contain ubiquitin. And also we were able to quantify the specific colocalization of ubiquitin with LC3 at the surface of the lipid droplets, or P62 and LC3 at the surface of lipid droplets. And we found that this association also increased following autophagy inhibition, indicating that this process is selective in macrophage foam cells. And so we use this model of aggregated LDL foam cells to isolate lipid droplets, and we wanted to identify the proteins that would be at the surface of the lipid droplets, tagging these for degradation. And we used THP1 macrophages for these experiments that we differentiated from the monocytes with PMA treatment for 72 hours. We loaded them up with cholesterol and lipid droplets by incubation with aggregated LDL for 30 hours. And then we compared two conditions. Chloroquine treated, we added the chloroquine in to inhibit lysosome degradation of lipid droplets. So this is what we use to inhibit autophagy and lipid droplet catabolism through this autophagy pathway, or untreated. And we reasoned that in the cells that were where autophagy was inhibited, the lipid droplets would be less degraded, and so there would be an accumulation of the tagging proteins at the surface. So that's why we did those two conditions. So first, we confirmed the purity of the lipid droplet fractions that we obtained. We can see that the lipid droplet fractions are selectively enriched in perilipin 2 as compared to the cytoplasmic fraction. We observed that these lipid droplets were devoid of GAPDH, calnexin, or LAMP1, which was only present in the cytoplasmic fraction. And we observed that LC3, both LC31 and LC32, so LC31 is the soluble cytosolic form of LC3, and this gets lipidated. It gets a phosphatidyl ethanolamine group conjugated to it, and then that allows the insertion of the LC32 in the autophagosome membrane. So we found the association of LC3 on the lipid droplet fractions. And we also confirmed the localization of perilipin 2, of ubiquitin, of P62, and LC3 at the surface of these isolated lipid droplets by immunofluorescence. So Sabrina was able to, after some trouble, figure out a way to get the isolated lipid droplets to stick on cover slips to perform this high resolution microscopy. So with the confidence that our lipid droplet fractions were pure, we proceeded to characterize the lipid droplet proteome. And what we identified, consistent with several other studies looking at the proteome, the presence of structural lipid droplet proteins, such as perilipin-2, perilipin-3, vimentin. We found the presence of machinery involved in neutral lipid droplet hydrolysis, such as this enzyme, cholesterol ester hydrolase, and ATGL, and also lipid droplet associated hydrolase. We observed several RAB proteins that have been previously implicated in regulation of autophagy, including RAB2, RAB6A and B, RAB11A and B, and RAB31. We observed several members of the ubiquitination machinery, including ubiquitin itself, which we were really happy to see. There was also association of this protein AUP1, which I'll talk about more later on. But this is an E2 ubiquitin ligase that has been previously associated to lipid droplets, and we think that this is the protein that's actually ubiquitinating the lipid droplets. We found the association of several chaperone proteins, including this TERA, a ubiquitin selective chaperone that's been previously implicated in aggregosome assembly and autophagosome maturation. We observed several lysosomal proteins, the cathepsins, also NPC2 and SCARB2. This is a protein involved in lysosome cholesterol efflux, and it's been implicated in the autophagy lysosome pathway. And we found the presence of several autophagy regulators as well on the surface of the foam cell lipid droplets, including COR1A, LTOR, a component of the lysosomal regulator complex that's essential for metabolic signaling. Several members of the 1433 protein family that have previously have established roles in autophagy, as well as DAP1, a negative regulator of autophagy, and HMGB1, which is a novel Becklin interacting partner that can promote autophagy. So with this list in hand, we also performed unbiased sequence motif enrichment to determine which protein motifs was most enriched in the group where we, in the cells where we included the autophagy inhibitor. And what we pulled out was the RAB, small RAB GTPase motif, ubiquitin, and 1433 proteins. Also, we found, we performed a gene ontology enrichment analysis to determine which of the pathways were enriched selectively in cells where we included the autophagy inhibitor. And we found that the top 3 pathways were response to lipid, vesicular trafficking, and regulation of autophagy. So what were the proteins that were enriched in the lipid droplet proteome upon autophagy inhibition? So we found a list of 37, which was, we were pretty happy. It wasn't too crazy, and it seemed highly specific. And amongst these, we found some previous proteins with previous known roles in autophagy, and others with no previous report of implication in the autophagy pathway. So of the ones that had previously that have previous described roles in autophagy, that includes HMGB2, which, like HMGB1, is a novel Becklin interacting protein that can promote autophagy. There was RAB8, which has been implicated in autophagy-dependent exocytosis, ApoD, which has been shown to be important to enhance lysosome function, and CIRB, which is an autophagy activator. Of the other proteins enriched selectively in lipid droplets upon autophagy inhibition with no known roles in autophagy, this included H2AY, a core histone protein, RBP7 with previous described roles in retinol metabolism, AGRL3, which has been shown to play a role in neuron guidance. And we heard a really nice talk last week about the importance of neuro guidance molecules in atherosclerosis by Wayne Orr's group, and GT251. So of these proteins, then we reasoned that if they were important in bridging the autophagosomes with tagged or ubiquitinated lipid droplets to be degraded by lipophagy, they may contain LC3 interacting motifs as well as ubiquitin binding motifs. So we asked which of the 37 proteins selectively enriched on the surface of lipid droplets following autophagy inhibition contained one or two of these motifs. We found several contained predicted Lear motifs, but in fact, only two contained both a Lear motif and a ubiquitin binding motif. And those were that neuro guidance molecule, AGRL3, and this other protein, GT251. So I'm not quite sure what these proteins, I hadn't really heard of them. If anybody knows something about these or any of the other ones that are selectively enriched on lipid droplets following autophagy inhibition, please feel free to reach out and we can chat about it. So now we wanted to test with this protein list in hand. We wanted to test, well, we needed to directly assess the role of these in mediating lipid droplet catabolism and cholesterol efflux. So we wanted to determine their functional role. Sorry about that. So we did this by designing a custom siRNA array, and we reverse transfected peritoneal macrophages, so primary mouse peritoneal macrophages onto the plate. And we had we made a list of our 90 top lipophagy candidate genes to knock down. And we had some controls, some positive controls in there and some negative controls in there as well. And then we loaded the cells with aggregated LDL and at the same time pulsed them with tritiated cholesterol to label the lipid, to incorporate tritiated cholesterol into the lipid droplet. So then we had our foam cells generated and we quantified cholesterol efflux to ApoA1 for 24 hours in these cells. And so here are the results. We can see that several genes reduced cholesterol efflux, and including several members of the autophagy pathway. So here, these are shown in pink. So you can see that the gene that most strongly downrate or reduce cholesterol efflux in these foam cells was LC3. This was included as a positive control. We know that it's essential to the autophagy pathway. Also, TFEB, ATG5, known regulators of the autophagy pathway, significantly reduced cholesterol efflux upon knockdown, as well as TFEB downstream genes, UVRAG, and also when we looked, there were several lysosomal proteins that reduce cholesterol efflux, including in PC2, SCARB2. Those were both at the surface of lipid droplets, and as well as prosaposin. There were several of the lipases that we identified on the lipid droplets in these foam cells where when we knock them down, reduce cholesterol efflux, including lipid droplet hydrolase, and also several chaperone proteins when knocked down, reduce cholesterol efflux, several of the RAB proteins, including RAB10, RAB2, RAB5, RAB6. And amongst the autophagy cargo receptors that we tested, we found that OPTN and NBR1 knockdown significantly reduced cholesterol efflux, but not BNIP3L and TAX1BP1 that have been identified as mitophagy cargo receptors. And both CalCoco2 and P62 knockdown reduced cholesterol efflux in 3 out of 4 experiments. experiments, although that was not significant. These two ubiquitin proteins, AUP1 and UBE2G2, both reduce cholesterol efflux, although this wasn't significant, but we observed a reduction of efflux in 3 out of 4 experiments. And these proteins that have not been described as lipid metabolism regulators in macrophages, previously RBP7 and H2AFY, also reduce cholesterol efflux. So of these cargo receptors, because OPTN and BR1, CACO2, and P62 all reduce cholesterol efflux, we next sought to look at their localization at the surface of lipid droplets. And we found that OPTN and NBR1, as well as P62, as we had previously seen with LC3, all localized to the surface of tagged lipid droplets. And I'm not showing the quantification that we did of colocalization of ubiquitin with OPTN, of P62 with ubiquitin, and NBR1 with ubiquitin, but all of these colocalized, and that this colocalization further increased upon autophagy inhibition, showing that it is specific. So there have been studies looking at microlipophagy in yeast. Obviously, yeast are easy to study, and they have ease of genetic manipulation. And so there have been previous studies looking at microlipophagy in yeast, and these have identified several of the core autophagy genes as being involved in lipophagy, but there have been no selective lipophagy genes identified to date, only a handful. NPC2 is one of them, actually. And so we used synthetic genetic array methodology to incorporate this well-established lipid droplet marker in yeast, FAA4, and also a vacuole marker, VPH1. And we incorporated these two tags into the yeast non-essential deletion mutant array library. So there's 4,000 mutants there, and that allows us to then visualize stat phase lipophagy. So after growing the yeast to stationary phase, this is 1 day after, you can see there are lipid droplets in these cells, and they're just kind of starting to localize to the vacuole. One of the advantage of turning to the yeast cells to look at the genetic requirement of lipophagy is that there's only one vacuole, so that's the yeast lysosome, as opposed to colocalizing, for instance, mammalian lipid droplets with mammalian lysosomes, of which we know there are several in the cytoplasm. So then you can see after 5 days of reaching growing the cells to stat phase, you can see the lipid droplets are being internalized into the vacuole here, and then you can compare the different strains. So this is the wild type strain, but then when you look at the strain lacking ATG8, which is the equivalent of LC3, which is absolutely required for autophagy, then you see that this vacuolar translocation of the lipid droplets is very much impaired. And so this is a powerful system for us to screen all of the genes of which there were yeast equivalents that we identified for the proteins on the surface of the lipid droplets. And we can look at this either by microscopy or through these FAA4 GFP cleavage assays. So that's that lipid droplet marker. You can see that following induction of lipophagy, there's breakdown of this protein as it's degraded in the vacuoles of the yeast, and then that generates a free GFP smaller product here that can be detected down here. And then we can look at the different strains. So here is the strain lacking ATG8, and you can see that the GFP cleavage is really abolished and impaired in other ATG strains, but not this ATG21 strain, which is ATG21 is specific to the CVT pathway, another specific type of autophagy in yeast. So we looked at, so we can begin to look at this by quantifying free GFP, so the cleavage of GFP in all the different strains, and we can classify this as low. So for in the case of ATG8, we scored this as being low out of 3 or more of 4 experiments. So here are the results for some of the strains that we've completed. You can see that there is a lot of impairment, for instance, in strains which lack PRK1 activity. So that is the mammalian AMP kinase. And we know AMP kinase is really important for autophagy activation. Also, you can see that several molecular chaperones, when they were deleted, yielded low lipophagy. Some of the Rab proteins, which we have we had identified at the surface of the lipid droplets that are involved in autophagy activation, such as Rab2 and Rab6, and Rab10 and Rab13. And also TFAB, UVRAG, yeast equivalents, the yeast equivalents lacking those genes showed low lipophagy. And then there were some strains that showed high lipophagy. So that would be these 2 strains, so VDAC, and when we looked at the genes, VDAC2 is an autophagy suppressor. So it makes sense that if we knock that out, then we would see high lipophagy. And Coro1A is a protein that blocks phagosome lysosome fusion, and so made sense that we would observe high lipophagy in that strain as well. So this model system has the potential of identifying both lipophagy inhibitors as well as lipophagy activators. So I'm sorry, my mouse is like dead, so I'm just gonna like charge it for 2 seconds. Okay. Yes. So so now, well, we have the DMA of 4,000 genes that Garrett, my PhD student, is systematically screening. Obviously, we have a kind of a bit of a targeted approach here, looking at the key pathways and the proteins that we've identified at the surface of lipid droplets first. But the goal is to get through and find a full list of genes involved in lipophagy. And so we also looked at this by microscopy, and we can score the translocation of lipid droplets inside the vacuoles. So here is wild type, and here is ATG8 mutant. So you see the vacuolar localization of the lipid droplets is reduced. And we also observed reduced entry for some of the key genes that we identified as proteins on the lipid droplets, such as this RNP1 delete strain here. That's the equivalent of the CIRBP protein that we found was enriched on lipid droplets upon autophagy inhibition. Also, we observed almost a complete lack of lipophagy in the UBC7 delete strain, as well as the Q1 delete strain. That is the equivalent of AUP1 in mammalian cells, and this is that UB2G2 ubiquitin conjugated enzyme. And then we could see instances where lipophagy looked to be enhanced, for instance, in this ATG15 delete strain. So this ATG15 is actually the equivalent of mammalian lysosomal acid lipase, which is the single lipase in lysosomes in mammalian cells that is responsible for the hydrolysis of cholesterol esters. So the quantification of this matched our visual observations. So we were able to score the vacuoles containing no lipid droplets, or the vacuoles containing less than 3 lipid droplets, or the vacuoles containing more than 3 lipid droplets. And you can see in the black bar here are the vacuoles containing more than 3. You can see that's greatly reduced in the absence of ATG8 and all of these strains, including the Q1, which is the equivalent equivalent of that AUP1 protein I was telling you about. And then in the instance of ATG15, where lipolysis, the degradation of lipid droplets, but not the vacuolar translocation, is impaired, then you can see that scores as having higher lipophagy, but that's because the degradation of these lipid droplets once they're inside the vacuole is impaired. So one of the things really interesting to us that we noticed when we looked at the list of proteins in the proteomics is that we noticed there were a bunch of chaperone proteins there on lipid droplets. And there was also this chaperone protein TERA that was previously implicated in lipid droplet clustering, not lipid droplet clustering, but agriphagy. And so one of the things that was kind of in the back of our mind when we looked at our lipid droplet microscopy was that sometimes there appeared to be lipid droplet clusters, but we kind of ignored those as like an artifact. But by seeing this list of proteins on lipid droplets involved in potentially agriphagy, we went back to the images and we carefully scored the amounts of single lipid droplets that were tagged by lipophagy. So this is shown here. You can see the single lipid droplets, and these are tagged with LC3 versus the ones that were clustered. So clusters of lipid droplets, this is an example here, would be lipid droplets that two or more lipid droplets stuck together in these clusters. And we found that these were also tagged with LC3. And, when we treated the cells with the autophagy inhibitor, chloroquine, we found increased tagging of both single lipid droplets as well as the clusters of lipid droplets, suggesting this was not, an accident. And then we quantified this, and you can see that both upon autophagy inhibition, tagging of both single and clusters of lipid droplets significantly increases, and that the proportion of lipid droplets within clusters in the cells is quite significant. Over 40% of all foam cell lipid droplets were in clusters. So we're not quite sure exactly what this means, and this is something that we're continuing to investigate in the lab, but is really, really intriguing to us. We could also find the presence of the clusters. We observe them by electron microscopy. So they're not an artifact of fixing cells and doing immunofluorescence. We see them also by live cell microscopy. When we label with Bodepi, we see the clusters as well there. So one thing that is intriguing in terms of the clusters and how they're being degraded once they're tagged for lipophagy by LC3 labeling is that autophagosomes are typically within the range of 0.5 to 1.5 micrometers in diameter size, whereas the clusters we measured were upwards of 2 micrometers in diameter. So how are these being internalized by autophagosomes? So that is unknown at this stage, but what we did observe was the presence of what seems to be wrapping lysosomes around the lipid droplet clusters here. And these wrapping lysosomes have been observed in the case of degrading mitochondria. So in fact, I was surprised to find that there's quite a bit of literature documenting both micro mitophagy and macro mitophagy. And so we're wondering if these two processes may be involved in lipid droplet catabolism in foam cells. In the liver, there is some evidence of microlipophagy as well by Anna Maria Corvo's group, which is quite interesting. And so this is also something that we're continuing to investigate, how the clusters of lipid droplets might be degraded. So this is, our working model. It's there's still a lot of unknowns, but we know that modified lipoproteins, such as aggregated LDL, trigger the ubiquitination of lipid droplets, and we think that the AUP1, E2 ubiquitin ligase, along with the UB2G2 ubiquitin conjugating enzyme, is involved in the ubiquitination of the lipid droplets, and that this ubiquitination of lipid droplets allows for the recruitment of autophagy receptors, OPTN, NBR1, P62, and that along with other proteins that will localize to lipid droplets, such as RAB2, RAB6, the HMGB1 and 2 proteins. This will allow and facilitate the phagosome assembly site for sequestration of potentially here single lipid droplets, since the clusters of lipid droplets that we observe would be too big for single autophagosomes, and potentially alternatively, these lipid droplet clusters may be degraded through an agrephagy-like mechanism. We don't think it's exactly agrephagy, because when we looked at yeast, where the TOLIP receptor that's required for agrephagy, Q5, was knocked out or deleted, that did not impair lipophagy. So it has to be something kind of agrephagy related, but not the same at all. So we're excited to continue the research on this and to find out. So I think I'll stop here. I had some slides on the potential role of lipophagy in sustaining beta oxidation, because this talk was really cholesterol focused, and the hydrolysis of lipid droplets would generate free cholesterol through the hydrolysis of cholesterol esters, but we also know that triglycerides are present in these macrophage foam cells, and that lipophagy would result in free fatty acid release that could then sustain beta oxidation production and anti-inflammatory functions in the macrophages. And this is something that we are testing now that we have several lipophagy factors that we've identified. So we can kind of test whether blocking lipophagy specifically could skew naive macrophages to M1 or prevent M2 polarization, and conversely test if activating lipophagy could skew naive macrophages to M2 or prevent M1 polarization. And we're really excited to have some of these tools now to really specifically test the role of lipophagy in macrophage foam cell processes, including lipid metabolism and inflammation modulation. And so our overarching goal in the lab is to understand the mechanisms of lipophagy, and the goal of developing some therapeutic strategies to selectively enhance this process to promote reverse cholesterol transport, and also potentially mitigate pro-inflammatory responses in these cells. And now that we have the yeast DMA to systematically identify new genes that can negatively or positively regulate lipophagy, that's a really powerful tool that we have in hand now. So I'd like to acknowledge all of our team members and also Via, who I forgot to mention, did all of the Western blots of the FA4 cleavage. So thank you, Via. And this is Garrett, who did the yeast work, and Sabrina, who did all of the macrophage foam cell work, and our collaborator, Kirsten Bates, which is the yeast person that has allowed us to perform all of these yeast experiments in her lab. And for the proteomics, Danielle Figges and Mathieu Lavallée Adam, and of course there's a bunch of people that I don't have time to all mention, but that I thank so much for their support. And I will take any questions.
Great, thank you, Mireille. That was so exciting. And I will take the first question before anybody else. Go ahead and raise your hand, of course, as per usual. So I have two small questions, I think. One is, in your, I guess in your siRNA screen, did you see anything that suggested increased efflux? So you showed us all that they inhibited efflux, but did anything that you maybe didn't show or anything, did you find any in the yeast? You seem to have found some that were sort of suppressors of autophagy, but did you see anything in cholesterol efflux that augmented cholesterol efflux?
Yeah, I do not believe that we did see positive regulators of cholesterol efflux. So I think we put all of the significant genes
They're all there. Okay.
So
Yeah.
no.
Okay.
But that's
just curious. It's interesting that everything was an inhibitor. Is it like siRNA? Not, so everything was an activator of autophagy that you found as opposed to inhibitors. Although if it was a lipid loaded cell, I guess they primarily, want to have loaded, like, anyway, it's kind of interesting.
Yeah, no, I think you're entirely right. And I was just thinking about that. As I was putting the slides together, for the proteomics, we really looked at the proteins that were selectively enhanced upon autophagy inhibition, but we didn't consider the ones that would be would go down, because that we did identify negative regulators of autophagy in our autophagy proteins that were there, like DAP1 is a negative regulator of autophagy, but we didn't look to see if that one was reduced. Yeah. So we need to go back and actually take all of that into careful consideration, the negative. We were just really, really looking for things that would allow for lipophagy to occur, but really focused on what would be enhanced at the surface of the lipid droplets. But that's an excellent point. Thanks.
Okay. Merit, I think you had your hand up first. I'm not sure, but please go ahead.
I don't think I was the first. Hello.
Okay, go ahead.
I don't think I was the first
afterwards.
One, but thank you.
Yeah. Okay.
Yeah, Mireille, that was a great talk. Yeah, no, it's very nice.
Thank you.
Thank you. When you're talking about cholesterol efflux, you're mainly talking about the ABCA1 dependent pathway. And basically the efflux that you see is entirely dependent on that pathway?
So I say it's ABCA1 dependent. It is preferentially, so when we initially did the characterization of autophagy efflux, we looked at, we compared wild type and ATG5 knockout and measured efflux to A1 or HDL in these two genotypes. And we found that like over 80% of the efflux to ApoA1 was reduced in the absence of autophagy, whereas only 25% of cholesterol efflux was reduced to HDL. And as we know, HDL preferentially, that efflux is mediated by ABCG1, whereas A1, poorly lipidated ApoA1, it's preferentially mediated by ABCA1. And that the reduction of 25% that we saw of efflux to HDL corresponds to the percent of lipid poor ApoA1 in HDL preps. So that is why we concluded that it was preferentially dependent on ABCA1. However, we haven't done the knockdown experiments to really establish, like in ABCG1 knockout, if autophagy has a role, because one of the other things that we're thinking about, of course, autophagy would supply free cholesterol to the ATP transporters, and these are upregulated in lipid-loaded cells. But as we know, there is basal efflux, and autophagy does impact that, and that's something that we're really curious about. So if we compare just efflux to BSA media, no cholesterol acceptors, in the autophagy knockout, there'll be like a 15% reduction in efflux. So where is that cholesterol go? And we think that maybe autophagy is involved in lysosome exocytosis. And that is something that, Sabrina has been working on as well, looking at that. And a lot of the genes involved, like mucolipin, when we knock that down in our siRNA array, that also reduced cholesterol efflux. And that is a key gene of lysosome exocytosis. And autophagy plays a role in that. So that's an excellent question. There's still a lot to be done to work out the efflux mechanisms through autophagy and lipophagy.
Okay, no, very interesting. And then I actually had one more kind of small question, because when you look at these lipid droplets that you had in your final slide, your schematic, of course, you have lipid droplets of a lot of different sizes. And then you were talking about the clusters, and you're talking about the small ones. But, when we have the course, like, I mean, working with ABCA1, ABCG1, we also observe these lipid droplets, and some of them are huge, some of them are really tiny. So is there also a difference in terms of like a gravity or other mechanisms that these are being degraded?
Yeah, so there's definitely a lot of, like, most of the work on the catabolism of lipid droplet has been done in the liver, so hepatocytes. And there's now kind of this this hypothesis in the field that first, large lipid droplets can be hydrolyzed by cytoplasmic lipid droplets, and then they can shrink down to a size that's small enough for autophagosomes to come in and sequester them. I don't know why the lipid droplets are clustering. I don't know if what regulates this. There has been implications of this protein HDAC6, which promotes lipid droplet clustering, and that's been observed. But I'm not sure whether this is through autophagy degradation. So I guess in a sense, it would be more efficient for the cell to do lipid droplet cluster catabolism, right? Because I mean, if you're going to be doing a bunch of little autophagosomes on tiny little single lipid droplets, that would be less, effective rather than having an agrophagy-like mechanism degrading a bunch of lipid droplets together in clusters. So we're really interested in we're teasing out the mechanisms between the two processes, and it's very intriguing because we kind of ignored these clusters and they're 40% of the whole lipid droplets in foam cells. So that was pretty crazy to us when we counted them. Yeah.
Definitely. Okay, well, thank you very much. Very interesting.
Thank you, Merit.
Can I go now?
Yes, go ahead.
Go ahead.
Great. Thank you. I'm from University of Calgary. It is really a great presentation. I enjoyed every bit.
Thank you.
I was intrigued by your EM data, actually. And my question is, once we are calling lipophagy or mitophagy, just focusing on the lipophagy, once it is entering to the lysosome, have you gone through the looking at the only in one lysosome degradation is happening or not happening? So is it only entering of the lipid droplets within the lysosome you are calling the lipophagy, or you watched over the time?
Yeah, so we did previously show that the catabolism required lysosomal acid lipase. So if we inhibit lysosomal acid lipase using lalostat, which is a selective acid lipase inhibitor, that reduced cholesterol efflux to ApoA1 to the same proportion that that autophagy ATG5 gene deletion did. And lalostat did not, or lysosomal acid lipase inhibition did not further decrease cholesterol efflux in ATG5 knockout cells. So yes, it does really promote the liberation of cholesterol in lysosomes through lysosomal acid lipase following the translocation into the lysosome. Question.
Just a short add-on on that, because in I work on the demyelination model, and in our field we know that cholesterol crystal is the result of accumulation of lots of lipid droplets. So I was thinking, if there is any inhibition or something is going impaired within the lysosome, do you see any of kind of those cholesterol crystal accumulation there?
Yeah, so that's an excellent question, and there's a lot of work first done by Babak Razani, who was first in Clay Semenkovic's group, that showed that in the when they had a macrophage specific deletion in mice in the bone marrow, they it was a lysine-cree ATG5 knockout in an athero model that led to increased inflammasome activation and increased crystals. And they did show that autophagy is involved in the inflammasome degradation, not the crystals themselves. But so, and also in the absence of autophagy, there is a lot of, there can be lysosome damage that is caused by like oxidized LDL loading because of the crystals, small crystals that are present. So yes, I think we haven't looked at like deleting autophagy in lipid loaded cells to see if there were like if the lipid droplets could crystallize, but that because I mean, in the lipid droplets, there's very little free cholesterol, so I don't think that could happen. There's you know, a very small proportion of cholesterol, but definitely in the lysosomes, like that does happen with atherogenic lipid loading, and then backing up the catabolism might back it up in the lysosome and then cause more. But that's definitely something that's interesting.
Thank you.
Thanks.
One more question, Zoe.
Okay. Oh, I have a quick question. So interesting in your M2 macrophage polarization. So you saw it's increased fatty acid oxidation. So I'm just curious, have you checked the glucose metabolism to see whether it's
So
going to decrease or increase glucose metabolism, because that's you.
Yes. So if you compare autophagy deficient macrophages, there is a decrease in fatty acid oxidation and an increase in glycolysis. Now, what we want to work out is, because autophagy, general autophagy affects so many aspects of metabolism. So we know, for instance, autophagy is involved in NCOR1 activation, degradation, and PPAR activation. So PPARs are really important for fatty acid oxidation. Also, autophagy regulates through mitophagy, mitochondrial function. So what we want to do is, now that we have some of the selective lipophagy, you know, tools in hand, genes, we can kind of modulate specifically that process and determine how lipophagy specifically, as opposed to bulk autophagy, is affecting macrophage metabolism and polarization. And yes, we're looking at glycolysis and fatty acid oxidation. Great question.
So I think this is all the questions. All right, Muriel, thank you for a fantastic talk.
Thank you.
It was a lot of great mechanism, and there's a lot of future of this resource. So we're really looking forward to hearing more about it.
Thanks for having me. Thanks, everyone. Nice seeing you guys. And I guess I'll stay on for the trainee coffee.
Yes.
Bye.
Bye, everyone. Have a good weekend. All right.
You still can stay behind if you want.
All right.
Oh.
Yeah, sorry.
Sorry, I'll make Mabruka a host.
Sure.
All right.
So it's an interactive trainee session, but everyone is invited to be on that.
Bye. Have a nice weekend.
I'll see you later, Miriam.
Yes. See you soon. Hi.
Hi. So very exciting seminar. And
Thank
when
you.
you mentioned smooth muscle cells, I was literally waiting to hear if there is a changes. Is there a difference between how smooth muscle cells handle lipid droplets compared to macrophages? So have you observed any differences between these two cell types?
Yeah, that's an excellent question. In fact, we just submitted a manuscript on this, of which Sabrina and Adele are co-first authors on. Yeah, we compared lipophagy flux in macrophages and smooth muscle cells that were sorted out from atherosclerotic plaques, like early atherosclerosis versus late atherosclerosis. And we find that autophagy is really selectively defective active in the smooth muscle cells.
Mm.
Not a surprise. We looked at some differential autophagy gene expression in smooth muscle cells versus macrophages. And there is a downregulation of certain autophagy activators and an upregulation of negative regulators of autophagy in the smooth muscle cells. And this occurs in smooth muscle cells that are lipid loaded. So one is TFEB. So when you lipid load with cyclodextrin cholesterol complexes, the smooth muscle cells, that actually downregulates TFEB. And so that's a critical autophagy activator that's activated in macrophages, but is downregulated in the smooth muscle cells. So, and then when we look at cholesterol efflux mechanisms, we don't really see, we see that if we activate autophagy in the smooth muscle cells, it doesn't promote cholesterol efflux to ApoA1 because ABCA1 is actually somehow less expressed than ABCG1. So there seems to be some potential for activating autophagy to promote cholesterol efflux in the smooth muscle cells that are lipid loaded, but that this would occur primarily through ABCG1. So the mechanisms are completely different of efflux in the macrophages and the smooth muscle cells.
So
Yeah.
have you worked on endothelial cells as well? Because those cells, they can become foamy as well.
Yeah, we haven't touched those, but honestly, it would be really cool to kind of sort out all the foam cells in the lesions, like, and then do like proteomics or spatial proteomics. I know that Holger Winkels was doing spatial proteomics in the in athero plaques. So I should actually see if he's published anything on this, because I mean, the last Gordon conference that we went to that was on immunometabolism, or atherosclerosis. I know he was, yeah, that was the last conference, and yes, he was talking about doing that. So I should follow up and see if there are lipid droplet proteins. I don't know if you can resolve like with the spatial proteomics. I don't think you can resolve like the organelles. So that would be kind of yeah, I think, but we need to pool like probably like 500 mice to get enough like lipid droplets. Because when we isolate lipid droplets from macrophages, we need, and Sabrina's like, yeah, like she needed to grow up like 15 centimeter dishes of each condition to like get enough lipid droplets for the mass spec. So yeah, I don't know how.
Time.
Yeah.
It's very hard job, right? How you can maintain the lipid droplets when you isolate them? Just do they resist to be ruptured when you with the processing? You have special media reagent? No.
Oh, so it's basically like an organelle, like you do a sucrose gradient and then the lipid droplets float up to the top because they're buoyant, because they're lipid loaded. So it's kind of easy. So whereas like all the other organelles are denser and like they go to the bottom. So the lipid, it's kind of like oil and water, right? If you put them, you mix it, and then the oil will always float to the top. So the lipid droplets are easy because they're at that top layer. And so you can just kind of get them, and then you basically precipitate the protein with acetone and you sonicate them to dissociate the proteins from the lipid, and then you precipitate the protein with acetone, and then you send that for mass spec. So it's not that difficult. It's just, yeah. Yeah, so have you been working on smooth muscle cells that are cholesterol loaded and stuff? Because I know a lot of people are having difficulty doing that. And if you need any like help, we've had our share of challenges with it, but now we have established a really good cyclodextrin cholesterol loading protocol for smooth muscle cells. And so if you need any, like Sabrina is doing it, and I think, like the human aortic smooth muscle cells like don't seem to load, and so but primary smooth muscle cells load well. So I think depending on the cell type, it might be one of the, so we're going to be comparing the human, because I know like Wayne Orr last week said that the human aortic smooth muscle cells don't load well, and I think Adil had even tried that and they didn't load well. So I'm wondering if there's like difficulty in the field in reproducing some experimental data, because like some people are using different smooth muscle cells, and Also like the cyclodextrin cholesterol source and FBS concentration, because they're really sensitive. If we load them in 0.2% like BSA, they die. So we've actually we incorporated FBS into the loading media, and they do much better, and they form these lipid droplets. But the lipid droplets are much smaller in the smooth muscle cells. They're different than in the macrophages. So
yeah, they don't collect it together like in macrophages. Yeah.
Yeah.
I'm interested in endothelial cells more because even Wayne work on smooth muscle cells. I'm endothelial person, so
yeah,
my work parallel to him. And I know endothelial cells, they form, they became foamy when you load them with the LDL, but it's very hard to process that.
Yeah. So do they express scavenger receptors? No, right?
They do. I think they do. Yeah,
Okay.
They do see 36, right?
Okay.
Yeah.
Yeah.
I have a question, but this is like not related to your science, which was amazing. Just because we have a lot of postdocs here, you know, something Katie told me a long time ago, but obviously it's super evident from your presentation, is you've done a really good job of integrating sort of like your PhD experiences and your postdoc experiences, but creating like a novel research program that doesn't really conflict with anyone else. And I'm just wondering if you have any tips for, you know, how to take all these bits of experience and create like a totally new and exciting research program.
Yeah. So I think one of the things, you definitely need to draw on your expertise, especially initially, because, you know, if I had proposed like in my first grant to look at lipophagy and smooth muscle cells and had no experience you know, looking at smooth muscle cells, then it would be a little bit like not super credible. Like I might be criticized for not having, like, the feasibility would be questionable basically. So definitely draw on your expertise, either from your postdoc or from your PhD, ideally from the two, and then create a niche for yourself. So lipophagy was in macrophages is not really studied by anybody else. I mean, there's a lot of lipophagy work, but not so much mechanistically. Like in yeast there is, but only like specific genes, and also you know, in liver. So in macrophages, like it could be like just another tissue from your mentor, right? So like Mabruka, you could propose something that is you know, your cell type of interest, endothelial cells, that pertains to overlapping interests with your mentor, where you'll have support with models and things like that, but that won't necessarily conflict with their interests. So, and like the communication is important. So I took one of the projects from Catherine's lab on ORP6. So this was an oxysterol binding protein. They found it was the most highly regulated miR33 gene, and I worked a little bit on it. But in fact, you know, the work that like they had started in the lab was on macrophages and hepatocytes. But I noticed, and you know, the tissue distribution, it was like more enriched in the brain, and a SNP in it was associated with Alzheimer's disease. So you know, I kind of asked Catherine if she had interest in that angle, and she was like, no, like you can totally take that. And so that's something that I branched out in the lab here. So, but I had previous expertise with ORP6 and cholesterol trafficking. The only thing that's new is I'm bringing it to the brain and now looking at the mechanisms in astrocytes. But so yes, it's a learning curve to like culture astrocytes and get familiar with these, but all of the other things, like I have the tools, like the flag construct, I have, you know, the experience to look at efflux and things like that. So it's not, so yeah, you definitely like should build on your, but then like go into a new direction. And, you know, ultimately, like you're going to have different directions for different, it's constantly evolving. Yeah, so I think like the brain cholesterol is something that I was really interested in, because like there's also not a lot of people working on it, and there's very few papers on astrocyte lipid droplets, and so my strength is lipid droplet, and so I could just apply that to like smooth muscle cells or astrocytes. And then I guess be open with your mentor. Like when I wrote my first grant, I asked Catherine to like review it for me, and she had excellent tips, but also that kind of showed her exactly what I was going to do. So there's no like surprises, like there's no, you know, and I've tried to like, so our first publication was like on the lipophagy, the work that I presented today that Sabrina led, and that was entirely without anybody I knew. It was only my group, and I'm really proud of that. I didn't even send Catherine my manuscript because I was like, I need to do this alone. Like I want to do it 100% alone. I didn't send it to any colleagues except our collaborators, and it's something that I'm really proud of because that like really signaled my independence, and that's what you want to show too, and that is important. And it's okay if you start on a project that is like a collaboration with your past mentor, but you really want to untangle yourself from them. Like I would love to collaborate with Catherine on so many things, and you know, she's offered help, like obviously, but I'm just like, I need to have a few independent, like completely independent publications from her, and then eventually I can collaborate again. Yeah.
Thanks.
That's really great advice. I'm gonna follow up with the publications. How important for us, so a senior postdoc, to have a corresponding author?
Oh, I don't think it's that important. I didn't have a corresponding author paper from my postdoc. And I think, I don't know, I mean, unless like it's evolving that I don't know about, but no, like the important thing is that you're first author on several good publications from your postdoc, but whether you're corresponding author or not, I don't think that really matters. And I guess like in Canada, it's different too, because we only apply for grants when we're independent and we have a position. Although that's different because like I think Adele did, but that's very uncommon to be a co-applicant on a grant. But yeah, it's really like based on papers. But I know the US is like you have to start like, you know, there's like the CDAs and things like that, like the path to independent grants and all of that. So but you know, it's better to start thinking about it in your lab now and start generating prelim. And that's what I did. I wrote my first grant application a year before I started my position at U Ottawa. And that really got me thinking about what I was going to do. And I did like have some preliminary data that I generated in Kat's lab before leaving. And then I went traveling in Asia for 3 months after my postdoc before coming. And I think that's like the last big trip I'll ever take in my whole entire lifetime.
I'd like to do a longer trip after my postdoc because
yeah,
I think it's a good idea.
Oh my God, if we can ever travel again.
So I'm going to ask you, what's the difference between finding jobs for postdoc in Canada compared to the US? You've been in the US and you went to Canada. Can I ask? Yeah. Is it like a difference or?
I don't know. I guess like, I'm not sure if there's that many. I think probably the hiring is similar. I'd say the US is just probably longer to have a real independent position, because like a lot of people work with like their grants but are still in like the same institution. In Canada you really have to move, and like then your lab's there and that's it. So I don't know that there are more positions in Canada than there are in the US. I'd say it's probably as competitive, but people probably start sooner and like just land on their feet sooner. Kind of like alone in an empty lab. But yeah, because like I feel like the postdocs are longer and in the US, and they're probably prepare you better. But I don't know. I'm not quite sure. What do you think?
I was going to ask a different question, but kind of related. But I'm just curious, like because I think we probably ask this to every person that comes. But how much time did you spend in the lab when you first started your lab? Because some people say absolutely 0 time, and then other people say, like Wayne Orr said, all the time. He lived in the lab for like 7 years. So I'm just curious.
So honestly, not that much. So I can't juggle it. And I was like that as a postdoc too. I'm not good at, I feel like it's a disruption. Like if you're writing a grant and you're in the lab, I quickly realized like I couldn't, and it might be like that. Like as a postdoc, I always had to like take a week or 2 off to draft a manuscript. Like I couldn't keep going with the experiments in the lab. Like it was just, you start getting into it, and if you're distracted every hour or 2 for a time point, it just really takes your mind off. And I just found I couldn't balance that as a PI. There's too many meetings and there's too many things in between, like meetings too. Like, how am I supposed to plan on 3 hours of tissue culture when like I have a thesis committee and like a meeting here and all these things. So I think as a PI, you have way more meetings. And so it's really difficult to find blocks of time where you can actually get into your mind and like do writing and things like that. So I can't do lab work. I miss it. And like I can step in if the lab's on fire, like if if somebody
animal
like
sacrifices.
yeah, animal sacrifices, like things like where it doesn't require, but like if I commit to an experiment, it's like 7 days, like often, right? Like if you do an eFlex experiment, you load at this time, you blah, blah, blah. And I was missing time points, and I just quickly stopped. I'd say I spent like maybe the first 6 months doing a bit of experiments, but mostly to like show people how to do things, like my technician and like a little bit Sabrina. Like if somebody needs help like reading their samples like in the scintiation counter, I can help them. And sometimes I'm like if they're waiting on somebody to show them on how to do a transfection, I might offer to do it. But the reality is like I also don't know where anything is anymore. So if you don't stay in the lab, you're incompetent, because you're like, I don't know where the pipettes are. I don't know where the falcon tubes are. I don't know where the filter tips are. I don't know, so it's just like your lab gets organized while you're in your office and you just don't know where anything is anymore. He's gone.
Like internet issues.
Yeah, but I can't balance the lab work and the you cut off. But like I was just saying, like you don't know where anything is anymore unless you're there all the time. Like people organize it around you while you're in your office, and then you don't know where things are. And so I think like it's great if you can do it, but I think it probably adds also to your workload, because you kind of like you're leading a double life. And I love, like I did a couple of time points recently in tissue culture, and I love it. It's great. It's like you're alone in the tissue culture, the silence, it's like peaceful, and I really miss it, but it's just not feasible with like the scheduling and it's so I admire people that can still do it, but at the same time, I feel like it probably way increases your workload. And I have a young child, and I also had a child like within a year of like starting my position. So that I was away from the lab, and that really like sealed the deal. When I came back, I was like, I don't know this lab. No, but like, you know what I mean? So also, I don't want to work like excessively my whole life. So I have to admit that I could work way more than I do, and I don't want to, because I've given it all in my PhD, I've given it all in my postdoc, but now I like have other interests such as my child. So I can't like the first year, like that I came back, I was like catching up and stuff, and I would work like all the weekends, and my husband's kind of like, not cool, yo. like he's always with like our daughter on the weekends. And so I think you have to, I was just talking about that with him the other night. You have to find out like your balance, because you know what, you could be this successful, you could be this successful, you could be this successful. It doesn't mean that you're not successful, but you can choose the extent to which you're successful. And I have ambition to be successful, but not the best. So because that would represent me burning myself out, and I'm already kind of like, I'm at my good capacity here, where if I take weekends, and if I really need to with a grant, then I'll work on the weekends. And sometimes I have like this like fantasy of working on the weekend, but then the weekend comes and I don't want to, and so then I don't. And then after, like, I'm like, Monday rolls around, and like, and so things just take more time than they should, and that's okay, because you don't have to work like 80 like hour weeks like your whole life. So yeah, that's important too, is like prioritizing too. Like, honestly, they tell you say no, and that is like the key thing, is like, so I really wanted to submit our manuscript, our last manuscript that we wrote, and I wrote it kind of like a little bit over Christmas, and like but there were the CIHR panels, and I was asked to sit on those panels, and I said no, and I feel bad, because it's like, you want to do service to the community, but like, do I need to do 3 or 2 CIHR panels a year? So now I said yes to the spring session, and that's fine, because we submitted our paper, and it's going to be like, I can do that one. But do I need to do 2 panels every year? No. So but it's hard to say no, because these are your peers. You want to like pitch in, you want to do everything. And same with peer review, where you're going to get like invites to review like 20 papers a week. And so do I want to review 20 papers a week? No. So like, sometimes if I'm not busy, I'll pick up more, but like, also if I'm busy, I have to prioritize myself. Otherwise I'm going to burn myself out. And if I'm burnt out, that helps no one. So it doesn't help my family. It doesn't help like my lab. So it's about working out like what you want, because there's no right or wrong in terms, I don't even remember your question. I'm just like rambling on.
This is a good, this
Enjoyed it.
is the best answer.
Yeah.
Yeah, I mean, I'm doing a lot of like data stuff, but I still feel this like pressure to go to the lab and do some experiment just to generate some data point. And I don't know,
Yeah.
it feels like, why did I should have just stayed home and like actually got some things done. So I don't know, it's kind of, that was a good perspective.
Yeah, it's really hard. Like, no. So what stage are you at?
I'm like a senior postdoc, I guess. I've been a postdoc for like 5 years. That's it. That sounds way too many years, but
No, I was a postdoc for 5 years. Some people do 3-year postdocs, but I don't think that's like the norm. I think most people do like, and you know what? It's a sweet time. Enjoy it. Like when you don't have to like worry about finances and like, where's the money like apply. Like, I mean, it's fun. I enjoyed all the stages, but don't feel like, are you starting to look for positions, or your paper is out?
yeah, I'm going to start looking mostly in the fall, I think.
And where are you looking?
In the US, but my wife wants to stay here in Michigan, and I'm willing to go anywhere, but she said if we leave, it has to be somewhere warm. So maybe somewhere warm.
Yeah. When I look at Ibrew's house, like,
yeah,
I know,
her
right?
backyard, I'm like, oh, it's like, it looks like a tropical paradise.
like
Yeah.
through the winter, it gets to look like that. It's not fair.
Yeah. So, but yeah, I think it's just like a personal choice, the lab thing. And also like the lab can be like an escape. Sometimes I see like people escape in the lab, where they like never go out and into the real world. And you don't want to do that either, right? Where you can't like, it's like, yeah.
Well, right now the lab is as close of a real world we're gonna get. So
Yeah. Yeah, it's true. It's true. Now, like before, it used to be like, you see people that are in all the time, like every single time you come in to do a time point at 10, they're there. You like come in early one morning, they're there, like they're always there. And you're like, what else are you doing? But it doesn't sound like that's your problem. So that's good.
Yeah. It's hard now when you have to schedule like every piece of equipment online and every room and all this stuff. I don't work that well with scheduling everything, and all the schedules are on different calendars. It's hard to keep it all straight.
Yeah, these are tough times, definitely. But like, I just feel